Hey everybody, in this video I am going to talk about chromosome abnormalities. So when we are talking about chromosome abnormalities, we are going to start with abnormalities that affect segments of chromosomes. And I'm using segments here because we have in the past talked about simple gene mutations in one gene. So that would be like the P color gene or the human albino gene. What other genes have we seen at? Drosophila eye color. So these were all single gene mutations. And when we're talking about chromosome abnormalities, we're talking about mutations that affect a segment of the chromosome. And in particular, we want to focus on segments that include or that span more than one gene. So when we're talking about mutations that span more than one gene, we're talking about a chromosome abnormality. So these can be deletions of chromosome segments, duplications, inversions, and translocations. Now we will take a look at each of these in detail, and then we'll talk about what happens when we have different numbers of chromosomes. So chromosome number differences. Now what do we mean by chromosome number differences? So essentially, right, humans have two sets of 23 chromosomes. Now, assuming everything is correct with each of the chromosomes, nothing's wrong with any of the chromosomes, what would happen if we had an additional set plus one set? So we had three sets of normal chromosomes. Another possibility with respect to chromosome number differences would be, instead of a whole new set, what about just missing one chromosome? Missing one chromosome, or having an extra chromosome. Now we'll take a look at all of these things in a video later on in today's playlist. But first, we're going to start with chromosome abnormalities. So damaged chromosomes, essentially, when a segment is lost or duplicated or inverted or translocated. So let's start with something easy. Let's start with deletions. So deletions of segments of chromosomes. And this one I think is relatively easy. So we can have a couple of, we can define deletions in a couple of different ways. So let's use this simple diagram of a chromosome. And remember, the long arms are Q, short arms are P, labeled P. And I'll define, these aren't genes, but these are different segments, different sections or segments of this chromosome. And these are going to help us take a look at the different types of deletions we may encounter in a chromosome. So two main types. We can have terminal deletions and interstitial deletions. Now with a terminal deletion, we are going to lose part of the end of one of the ends of the chromosome, and with an interstitial deletion, we will lose a segment from the middle, not from the ends, but from the middle of the chromosome. So let's see if I can copy and paste this. Copy and paste this diagram. So we have one here. Oops. I got greedy. Okay, let's see. See if I can make another one. Perfect. Okay, now for the terminal deletion, let's assume, now remember this chromosome is one long piece of DNA. Let's say it hasn't undergone DNA replication yet in S phase. And let's say the deletion happens right here. And for the interstitial deletion, let's say we have 2 DNA breaks at these 2 arrows here. So we have a DNA break over here and over here. With this one, we have 2 DNA breaks. So let's say when the DNA is repaired, what happens for the terminal deletion is the DNA is not repaired. We just lose that end. A and B. The A and B segments of the chromosome are lost. And okay, so here is our chromosome with a terminal deletion. It's missing segments A and B, and that could be bad, right? Because all of those genes in segments A and B will be lost. Or they'll be present in single copy, because let's say this is a diploid organism that the homolog still has segments A and B. I should diagram this a little shorter. With respect to this chromosome up here. Okay, so let's say these 2 DNA breaks occurred, and when the DNA was repaired, that segment C and D was lost. So this B segment here was joined to E. Okay, so here we lost segment CD over here. Okay, so this is an example of our interstitial deletion over here. And this is an example of our terminal deletion over here. Now, in introductory genetics classes, it's very common to talk about a human syndrome called Cri-du-chat, cry of the cat, which is caused by a deletion, a terminal deletion in chromosome 5 of humans. So chromosome 5 terminal deletion. Now, the hallmark, I guess, clinical feature of this syndrome is the newborn cries like a cat. So there's a cat-like cry, and I think this cri du chat is French for cry of the cat. And if you want to hear that, I think some parents put a video of their infant with cri du chat syndrome on the internet, and I have the website here. Where did that go? So you can check the notes for the website. But it's on YouTube. So youtube.com/watch lowercase v equals uppercase T, uppercase Y, uppercase Q, lowercase r, lowercase z, uppercase F, uppercase A, uppercase B, uppercase Q, uppercase H, uppercase Q. So if you don't have the notes with you, You can just type that in, and you should be able to go to a YouTube video where you can hear what this cat-like cry sounds like. So there are many other clinical features. Some of them can be pretty severe. That said, there are others with this syndrome who are highly functioning. So there's a wide range of symptoms and severity of symptoms. 90% of infants were born with this syndrome. So the source is a de novo mutation, which means neither parent has this terminal deletion of chromosome 5, but it happened during gametogenesis. So formation of the sperm or the egg that led to the child. So 80% of these de novo mutations are thought to occur during spermatogenesis. So, which is kind of interesting. Now, researchers have found or narrowed down the location of the regions responsible for Cri-du-Chat syndrome. So let's say this is chromosome 5 in humans, and this is the P arm, and this is the much longer Q arm down there. Now, researchers who I guess cytogeneticists, these are geneticists who study chromosome structure using microscopes and things like that. So they tend to use a stain called Geisma. So Geisma staining of chromosomes is a very common technique in genetics, especially for cytogenetics. So with Geisma staining, we can get these things called G-bands to form on the chromosome. So when you look at the chromosomes under a microscope, you can see these bands that occur in the chromosomes by according to the amount of stain the chromosomes take up. Now, certain parts of the chromosome that are rich in A and T nucleotides, so adenine and thymine nucleotides, so AT rich regions are thought to take up more stain, so they'll stain darker. And G and C rich regions tend to take up less stain, so they will stain lighter. And humans are, our genomes, our chromosomes are like 99.9% identical to each other. So each chromosome, each chromosome 5 in a normal human should have the same banding pattern, especially if banding is related to the A and T richness or G and C richness of a certain section of a chromosome. And because of that, we can use G-banding to quickly analyze the structures of chromosomes in an individual human, and can check and make sure all those bands are there to see if a human has, a normal set of chromosomes, not missing any segments or not having any segments duplicated. And so what do these bands look like? So let's say we have two dark bands here and a dark band here and a dark band here. So every human, as long as we don't have duplications or deletions, should have these same bands on chromosome 5. And they have certain numbers. So this one right here would be 5Q. I believe this one is actually 5Q11 because this is on the Q arm right here. And this one, we're going to call this one 5P11. And where do these numbers come from? I'm not sure where those numbers come from, but the first band on chromosome 5, closest to the centromere in the Q arm, is called 5Q11. And the band closest to the centromere on the P arm is called 5P11. Now, I have a diagram of this. I already sketched it for you. And now, I'm not an expert in G banding of human chromosomes. So this is, I'm using Wikipedia here. I'm using the example on Wikipedia, and I use Wikipedia a lot, and I've never seen anything completely inaccurate on Wikipedia. I think it's a great source for biology and genetics. So, but this is something I'm not really that sure about, but I'm pretty sure, it's probably accurate. So, these are the major G bands. These are the major, where's my marker? These are the major G bands on the P arm of chromosome 5 in humans. So, like I said, here's the 5p11 down here, and there's a lighter 5p12 up here. And then this one right here, you can see how it doesn't take up as much stain. Now that's going to be 5p13.2. I did not put the 5p in there, but technically this name is actually 5p13.2. And then we have 5p13.3. So the regions between the bands that don't take up stain, in between this, the actual darkly the actual bands have numbers. So you can make use of the stain, not just by which regions take up the stain, but which regions don't take up the stain. So why am I pointing all this out? Well, it turns out researchers have narrowed down the locations of chromosome 5 that are responsible for the clinical features of Cri-du-Chat syndrome. So it turns out that 15.3. Let me say that again, sorry. Chromosome 5, arm P, band 15.31 is a segment that is responsible for the cat-like cry of Cri-du-Chat syndrome, and 15.2 is responsible for all the other clinical features of the syndrome. Okay, what that means essentially is that an individual that has the cat-like cry and other features is missing or has the terminal deletion occurring somewhere in 15.2. An infant that just has the cry has the terminal deletion occurring, closer or within 15.31. So I thought that was rather interesting. So what we can do is, if I were to ask you a question, now, let's say you're a physician or OBGYN and you deliver an infant and you can hear that characteristic cry. So you order a karyotyping analysis where they look at the chromosomes of the newborn, and you see something like this for the two chromosome 5s in the infant. Oh, my Q arm is too short, isn't it? So you might see something like this. Okay, we'd expect to see a terminal deletion here in chromosome 5. And if that deletion occurs in 5p, right? 15.3.1, then that infant is probably only going to have the cat-like cry feature of the syndrome. If the deletion occurs or occurred in 5p15.2, we can expect a more severe form of Cri-du-Chat. Of course, I didn't put all the bands in here for these chromosomes here. Okay, I think we get the point of how Gbanding can be used to quickly look at chromosomes and see what parts of chromosomes have been deleted. And we can also see how Gbanding can be used to diagnose certain disorders such as Cri-du-Chat syndrome. Now, let's take a look at, just a quick look at duplications. So, I have some notes here. Okay, so let me diagram a chromosome here. And some segments in here. A, B, C, D, E, F, G, and H. Now, duplication is relatively easy. Okay, so any of these segments, one or more of them, will be duplicated if a chromosome abnormality is involves a duplicated segment. So let's just say segment C was duplicated. Okay, so we have two segments there, a duplicated segment. Now, is this a problem? So we need to consider a number of things to know if this is going to be a problem. Usually, often, especially with humans, well, I shouldn't say that, because it turns out that scientists are learning that humans have, these are very common in our genome, especially small duplicated segments. So the problem is when the segment is long, when the duplicated segment is long, we can run into trouble. So the longer the segment, usually the longer the segment, the bigger the problem. Okay, why? Why would that matter? So longer segments, usually, it's kind of intuitive, right? Usually a longer segment would have more genes than a shorter segment. So if you have a long segment that was duplicated, now every one of those genes is going to be present in higher numbers than it was before that segment was duplicated, right? So, and if you have more copies of a gene, usually that means more gene product. And too much gene product for too many genes, or too much gene product for many genes, you're more likely to have problems. So a lot of things we need to consider when determining if a duplication will cause problems for the organism. A segmental duplication will cause problems for the organism. Essentially, the longer the duplication, the more likely we are to see problems. So another classic example here that we should cover, now that we're taking a look at duplications, involves the Drosophila bar gene. So the bar gene affects the phenotype of the eyes of Drosophila. Now this gene is found on the X chromosome, and it's found in a region of the X chromosome in Drosophila called 16S. Now, so normal Drosophila females might look something like this. So let's say this is an X chromosome. Drosophila is a diploid organism, so I can diagram another X chromosome in the female. Female Drosophila have two X chromosomes. So here's the 16S segment present in single copies in both of these X chromosomes. And these flies would have what we call the wild type phenotype. And for the bar gene, there's only one copy, and they have wild type alleles, and they have the wild type phenotype. So we're going to name each allele B+ when it's present in one copy and it has the normal sequence. So it will have normal eyes. Now, if that segment is duplicated on one of the chromosomes, we have 2 copies of that segment. As a result, there'll be 2 copies of the bar gene, and that's going to be those 2 copies, those 2 segments of 16S are typically referred to as the B allele, which is kind of confusing, right? This B allele, it's dominant too, really refers to 2 segments, and there's 2 copies of the bar gene in there. This chromosome over here just has 1 copy of the bar gene, so we're going to refer to that as B+. So these flies have the bar phenotype. So the eyes are going to look like little bars. And we have another possibility here called the double bar mutation. So essentially, if we have 3 copies or 3 16S segments, so a triplication of that segment, we get what is called the double bar allele. So normally when we're talking about alleles, we're talking about different versions of one gene. But in this case, the allele is actually refers to the three segments of 16S. So again, these are dominant over wild type. And so this is going to be the double bar phenotype. Now these phenotypes are resulting from too many copies of the bar gene. So you have too much product and you get abnormal phenotypes. So this is an example of how a duplication can lead to an abnormal phenotype. And okay, what do these look like? Well, I have some diagrams down here. Let's see if I can find it. Let's copy. And go back up here. Perfect. Okay, so if this right here is the normal phenotype, nice big red eyes, here's the bar phenotype where the eyes are like little bars, right? This is like a little, instead of being a nice round shape, it looks like a little bar. And this is the double bar phenotype, where the eye looks even more like a bar. And when we say bar, we mean like a lead pipe type of bar type thing. Okay, so that's an example of how a segmental duplication can lead to an abnormal phenotype. And that's just one gene in the segment. So segmental duplications aren't always bad, right? And so there are a number of things to consider when you're talking about duplications, right? The length of the duplication is really important. The longer the duplication, the more likely the more likely the duplication is to be problematic. But let's take an example right here of a relatively small segmental duplication where we have a segment of a chromosome with 3 genes. So gene 1, gene 2, and gene 3. So let's say this duplicates, this segment duplicates. And sometimes duplications can occur where they don't, the duplicated segment moves to a new part of the genome. And sometimes, usually I think the duplication starts where it occurs side by side. And that's a tandem duplication. So the duplication is found right next to the parental segment that was duplicated. Okay, so now what we have is gene 1, gene 2, and gene 3. And over here we have gene 1 prime, we're calling this. Gene 2 prime, these are the duplicated copies. Gene 3 prime. Okay, now that we have these essentially 2 copies of each gene, and let's assume that this duplication has no effect on the organism, no noticeable effect. So what can happen now is we can have mutation and evolution of new functions. For example, let's say genes 1, 2, and 3, they were critical genes, and they have a function that must be performed in the cell. So any mutations in those genes would eliminate that function, or could eliminate that function if they created like a null allele. Or any mutation that changed the function slightly could have a negative impact on the organism. But now, after this duplication right here, the cell can use one of these copies. Let's say it uses the copies on the left. Doesn't have to occur this way, but the copies on the left still do their normal functions, are still used for the normal functions. But these over here now, okay, this evolution is free to, mutate these and try out new things, right? A mutation that changes the function slightly, and if that function offers a benefit to the organism, well, okay, well that would be a good thing, and then maybe that gene would, that organism would go on to produce more offspring, and that duplicated gene with the new function would spread through the population. More often than not, mutations in these genes lead to null alleles. So they inactivate these genes and create what we call pseudogenes, and we'll learn more about these later. So pseudogenes, these are genes that result from duplication and they're no longer functional. They acquired mutations and they don't function anymore. So segmental duplications can generate genes which can mutate to develop new functions, or they can mutate to become pseudogenes. So to summarize, segmental duplications aren't always a bad thing. So they can provide genetic material for evolution to create genes with new functions. Okay, so that's it for deletions and duplications. In the next video, we are going to take a look at inversions.